about GOLiath. This is a piece of software for performing gene set enrichment analysis. And I'm also going to have a look at a couple of other tools for carrying out this type of analysis as well. So functional analysis on gene sets is a very popular thing to do. Often at the end of an analysis, when all the data has been processed, it can be a really useful thing to do to help interpret results from experiments, hopefully neatly tying up experimental results and perhaps giving suggestions for future work. So the type of functional analysis that I'm covering today essentially involves taking a set of genes, preferably around a few hundred, from the output of an analysis. They may be genes that have been identified as differentially expressed between conditions or the output from another statistical test. And then we test these genes to see whether they are enriched for particular biological functions. There are plenty of freely available web applications to do this kind of analysis. And most of these tools offer something slightly different in the way of visualization, the statistics used, or the range of data sets or annotations used as the set of functional categories to test against. I'll come back to this again shortly. But for now, I've picked three tools here that I've put the same set of 200 genes through. So I will just share my screen now. So okay, so these are 200 mouse genes that I've just run through an analysis. This is gProfiler, which is a nice tool for doing this kind of analysis. And I've already run it, so these are the results that it's produced. And we get this quite nice little plot down here, where these little circles are functional categories that have been identified as being significantly enriched in this set of genes. So if I mouse over these circles, we can see information about the category. On the y-axis, we've got an adjusted p-value, so this is the measure of significance. So by the higher the number, the more significant category is. And then along the x-axis, we've got the different types of functional categories. So a lot of these are from gene ontology. We've got the three different subcategories of gene ontology. We've got molecular function, biological process, and cellular compartments. And we can click on these circles and see a little bit more information in the table. So that was the default first view from gProfiler. We can also have a look at the more detailed results. That's going to come on, which shows some more information in a table. So we've got more information about all of the categories, and we can have a look at some statistics things in there. I will just have a look at another tool now that I've also run that same set of 200 genes through. So this is GOLiath, and GOLiath just tests against gene ontology categories, and it has a slightly different layout for the results. So I can go through the different subsets of the ontology categories. So this one at the moment is on biological process. The molecular function results fit slightly better on the screen, so we'll just have a look at that one for now. So this kind of display shows the hierarchy of the gene ontology terms, and it shows the statistical significance of the results by colour. So these darker boxes are more highly significant. And if we scroll down, then we get more information here, just like in gProfiler. And then one last tool that we'll just look at briefly is Panther. So Panther also just uses the gene ontology sets. And in this case, I've chosen to use the molecular function subset, just like in GOLiath. And this is the default view that Panther shows. We've got all some information in there. They do offer some plots things, but this is the view. So just head back to gProfiler. So there are a couple of reasonably simple things that can make a big difference when doing these analyses. The first is a background list. So we have our a set of query genes of interest here. I've got a couple of hundred here. And then we also have a larger set of genes that our query genes have been selected from. The larger set may be an entire genome, if it was a genome-wide experiment with values across the whole genome, or it may be a subset of the genome for which we've got measurements for. So it might be that only certain genes are expressed under the experimental conditions, and it's all those genes that are expressed that we should use as the background set. So gProfiler just assumes that we'll use a whole genome background unless we tell it otherwise. GOLiath makes us put in a background, and Panther lets you do either way. It does provide one, but it also allows you to upload your own. So putting in a custom background can make quite a difference to the results. The other thing that can make quite a difference is looking at the size of the set of functional categories that the genes are tested against. Or indeed the sources that are used. So Panther uses gene ontology. You can choose from which subset from a drop-down list. And GOLiath is similar as well. It tests against all the gene ontology terms. gProfiler offers a few more options. So they also, then the bulk of the functional categories are from gene ontology, but it also offers, it can use other sources as well. So we've got some pathway data and some transcription factor and phenotypes, proteins as well. So we could upload a custom GMT file, which is generally what is used to, that includes our set of functional categories. So this is a small example of one of these, containing functional categories. So what we have in here is a name of a functional category, and then we'll have the source that it's taken from. So this is a Go biological process, gene ontology biological process. We've got its name again. And then we have the genes that are or have been annotated as being linked to this category. So in this case, there's only 6 genes in here. You should be able to see that some of the categories are much larger. So this one's got quite a few more genes in there. Some of the other categories, some have only got one gene in, and some that aren't on here have got thousands. So doing the statistics on these different sizes of categories makes it a bit interesting. So for example, so if one of your query genes happened to be in a category with a very small number of genes, then statistically that's going to come back as very significant. You wouldn't expect your gene to be in there by chance. But there are many thousands of these categories. We also see very large categories coming up in results a lot. So for example, in gorilla, we can see this protein binding category has been identified as very significant. But I think that one has about 9,000-10,000 genes in it. So it may be statistically significant, but really not particularly useful when it comes to the biological interpretation of the data. So if half the genes in the genome are in a category, then it's not particularly going to tell us very much about our set of genes there. So most useful sets biologically are probably not going to be those that are at these extremes. They'll be the ones that have more than a handful of genes, but not thousands. So what then happens in the analysis is that for each of the categories in the file, we check how many of the query genes are found within this category, how many of the background genes are within the category, and use a statistical test to determine how likely it is that the proportion of query genes in the category is due to chance, or whether we can be confident that there is real enrichment there. So that gives us a p-value, and as there are generally thousands of these tests carried out, a multiple testing correction is applied, which gives an adjusted p-value. And we can see these in the results. So they're p-values and adjusted p-values, or false discovery rates in some cases, vary slightly between the different tools. And this depends on the statistics used and the sets of functional categories that have been used. But we do tend to see the same or very similar categories coming up across the different tools. So through doing lots of data analyses for people on lots of different data sets and seeing many sets of results from these programs, we'd often find that we get very significant p-values. So you can see these ones are very highly significant. And so the results can look very promising and exciting. But we also noticed that quite often certain categories seem to keep coming up that we just didn't necessarily believe meant very much biologically for the data set in question. And this was really frustrating, as they were looking like great results, and we couldn't really explain to people why we didn't believe that some of the categories should really have been there. So the results of this here are perfectly valid statistically, and there's nothing wrong with the tools. It just didn't always seem quite right, but we couldn't give a valid explanation as to why. And we really wanted to find out why some of these categories appeared. So the sets of genes that we were generally putting through these functional enrichment analyses were from sequencing experiments, often RNA-seq or ChIP-seq or similar. And we know that throughout all steps of library preparation and the analysis of sequencing data, that quality control is really important, and that biases and artifacts can be introduced into the data at various points throughout the process. We've got lots of quality control tools for examining data at various stages, looking at FastQ data, map data. But when it gets to the end of the processing and analysis, and we've just got a set of genes out at the end, if we're just given these genes, then we don't really have more information than that to go on. So we know that it's easy to get GC biases in the data through PCR in the library prep. We know that when we perform statistical tests to identify differentially expressed genes, the lengths of genes or expression level can confound the results in ways that aren't obvious if you're just looking at the output of a pipeline, and that various other biases can be introduced as well. So we thought it would be interesting to generate sets of biased genes and to run these through a functional enrichment analysis to see whether we got any significant results. So we generated sets of genes biased by GC content, so low GC genes and high GC, and so genes biased by length, so short genes and long genes. It's still for mouse at the moment. We've done this for mouse and for human. And also sets of genes where there were a high number of transcripts or splice variants annotated in the genome. Genes that were closest to randomly generated positions in the genome. And we've also tried taking genes from just one chromosome. And for each of these different categories, about 200 genes were randomly selected and run through a functional enrichment analysis. This was repeated a couple of hundred times to see which functional categories came up the most frequently. And for some of these biased gene lists, the gene set enrichment analysis produced lots of highly significant results, and for others, not so many. But we essentially ended up with a list of functional categories that were potentially suspect. As another category, we also downloaded a load of public mouse RNA-seq datasets with 2 biological replicates and did a fairly standard RNA-seq analysis to identify genes that changed the most between the 2 replicates, so where we wouldn't really expect to see differences, and then ran these genes through an enrichment analysis to see whether any particular functional categories were identified. And this did actually produce quite a lot of categories to add to our list of potential suspects. So these we then used in GOLiath. So there's a link to GOLiath from the Abraham Bioinformatics projects page down here. And GOLiath is similar to the other tools that we've seen, where you can paste in a set of gene names and perform a functional enrichment analysis. So I'll use the same set of genes that I used for the other tools, and at the moment GOLiath just supports human and mouse genes. So I talked a bit already about how the category sizes affect the results, and said the most biologically interesting sets are probably those with a few hundred genes. So that's why we did have those defaults set there. I'm just changing these for now so that we get more similar results to the other tools. I'm just going to leave the background list as a genome-wide set. So most of the results columns will be similar to the ones we've seen in the other tools, where the first column is the name of the functional category. The GMT file, so the one like this, that is used in GOLiath is from the Beta lab. So they collate functional information and categories from a few different sources. So the main sets are the gene ontology, but then they've also got some pathway data sets in there, and they keep these nicely updated. So this is, so GOLiath's finished now. So as I said, most of these columns are similar to the other tools that we've seen. So we've got the name of the category, functional category here, the source of the category, so these are gene ontology sources, this is molecular function. The query count is the number of genes from the query set that were in the functional category. The background count is the number of genes from the background set that were in the functional category. And in this case, I didn't put in a custom background, so it just uses a genome-wide background. Then we've got our measure of significance, a false discovery rate, and an enrichment score. And then this last column is where we make use of the set of potentially suspect categories. So GOLiath checks the category name, and if it matches one in the list, then it gets flagged up. So this first category, the cytoskeletal protein binding, theirs came up when we ran sets of biased genes through functional enrichment analysis. So in this case, this category came up when there were genes biased by high numbers of transcripts, long genes, and also in the public data, as did most of these other categories here. So this was a bit of an extreme example. The 200 genes that I ran through this analysis all have a very high number of transcripts, splice variants. So a lot of them are long. And so some were also in the set of genes that were closest to random positions in the genome. And some of the categories here were also identified when analysing the public RNA-seq data. So although this is more extreme than you should ever see for a normal set of genes, it does show that we can get what looks like a lot of highly significant results from a very biased gene list. So if we have a look back in gProfiler, we should see some of the same categories coming up. They're going to be slightly different due to the sets of functional categories used in size limits, etc. But if we have a scroll down, we're trying to think about whether these results look interesting. You might think some of these categories are a little bit broad, so binding is a massive category that's going to involve a very high proportion of the genes in the genome, so perhaps that doesn't look so interesting. Organelle also is a very, very broad term. But we've also got a load of transcription factor data that could look quite interesting, and some phenotypes around here that are also pretty significant. So yes, this does all look quite interesting, even though this was just a set of genes taken from genes with a high number of splice variants. So if we go back to GOLiath, there are a couple of other things we can look at here just to perhaps try and understand a bit more about what's going on with our data. So we've got a properties tab just here, which gives us a bit of basic information about our query. We've got a couple of plots here for a bit of quality control information. So this one is showing the distribution of lengths of gene for the sets that we've used. So in black, we've got the distribution of the lengths of the background genes versus the query. So our query genes are generally longer than a lot of them are longer than background genes. Something interesting going on with the distribution of the GC content of our query genes too, but we know this was a very biased gene set, so which we could also see in this plot down here, which shows the distribution of genes on chromosomes, the genome. So for our background set, genome-wide, the proportion of genes on each chromosome is reasonably spread out. So whereas my query set are mainly from genes from 1, 2, and 3 and chromosome X. So this was a very biased gene list. So hopefully we wouldn't see results like this for a proper analysis. But if I just go back to the home page and just going to run an example through again with some other example genes. I'm going to increase the category size to 5,000 here just so that we can have a look at what comes up. So I've got a question here saying we're using the same gene list for these different tools. They give different scores and results. And which one is the best, I think is the question. Gives perfect pathway list and enrichment score. And it's very difficult to say, to just pick one tool and say they are the best or they are right, because they're using different statistics, and they're using different sets of functional categories to look at. Okay. We'll come back to that. Okay. So so this set of query genes produces some different results, and I can filter the. We can always filter for the size of the category afterwards when we've got these results. So actually, I'm going to filter the maximum category size now to 500, and now we've got quite a few. We've got quite a few categories here that aren't being flagged up as potentially biased. So a lot of the suspect categories do have a high number of genes in them, so we can filter out quite a lot by just removing those large categories. And really the most biologically interesting categories probably aren't going to be the broad ones that have thousands of genes in. 13,000 genes for the binding set is probably not going to be that helpful in the analysis. But we can have a play around with this size afterwards. So these flagged categories here don't mean that the result isn't valid. The flags are just there to show that the fact that this category is coming up may be So this one in particular, signaling by interleukins, is flagged up as with a high transcripts flag. The fact that this is coming up may be due to there being lots of genes with high numbers of transcripts in your data. This may be perfectly fine and something that you'd expect for your data, or it may be something that you should look into further. So where we've got the public data flag coming up, that's showing that that category came up a lot when analysing lots of other data sets, and we wouldn't really have expected to get significant results. So we might want to be a bit more wary of that category. Where we've got lots of flags coming up without an obvious reason for the data in question, then we might want to investigate the data a bit more and the processing and analysis that's being carried out. So we've also, so in summary, and I will come back to questions. So GOLiath can be used as a standalone tool for normal gene set enrichment analysis and to help to identify any issues in the data or to provide reassurance that there don't seem to be issues, and to help identify any functional categories that we may want to be wary of. And it can also be used as a complementary tool to others available, such as the ones that we've looked at briefly. So this is still very much under development, so any issues, then let us know, or feel free to make improvements on GitHub. So I'll just, that's mostly what I was going to cover for using the tool. I'll just answer a couple of questions that we've got in the chat now. So defining potential bias was an interesting one. So we've got, so there are some documents where we've kind of explained how these were generated in more detail. So so there are different parameters for the different sets. So for example, on the length biased genes, we took genes that were the longest 10% of genes in the mouse genome here, and ran those through, or ran sets of 200 of those through the functional enrichment analysis, and looked at the categories that were identified as being significantly enriched. And this was then repeated, and then we took a threshold for the number of times the category came up, and if it was above this threshold, then it got added to the list of potential suspects. So at the moment that threshold is fairly arbitrary and can be subject to moving around really, but as with all of this, it's pretty difficult to set thresholds in this. But yeah, these documents, I'll go through those a bit more. So the public data, so the public data sets were, there were a few hundred mouse RNA-seq data sets from ENCODE that had 2 biological replicates, and so we just did a differential expression analysis on those. So we wouldn't really expect to see much difference between those 2 data sets, but we took the genes that were identified as the most differentially expressed and ran those through functional enrichment analysis to see whether we got significant results. And we did for quite a lot of those. And again, if the categories came up a certain number of times, then those were added to the list of potential suspects. So it's not saying that the category itself is bad at all, just that perhaps it seems to be more likely that this category will be identified when perhaps it shouldn't have been biologically. So it might just be that there are genes in that that are very easy to easier to select for in our analysis, whether we mean to or not. So, so yes, that was the public data. The public data had only been done for the mouse data set. So we've got potential suspects for human data, for the high transcripts and long and GC content and chromosomes, but we haven't got that the public data for the human data for human data sets yet, but we should process that and add those on. So yeah, I think that's pretty much what I was going to cover. Packages, there's a question about packages performing Go enrichment analysis using R scripts for yeast. Don't know particularly for yeast. I know gProfiler does have, and they do provide an R interface and scripts, which we had used. That seemed to work quite nicely. I think there are a few of them around. I mean, the actual analysis isn't too complicated. It just can change depending on the functional sets that are used. But hopefully, you should see that with whatever package or tool is used, if the results are highly significant, then they tend to be very similar across the different tools and packages. How differential expression analysis with this tool? Not sure I totally understand Dave's question. So when I was talking about differential expression here, so the differential expression analysis was further up in an analysis pipeline, and then these kind of tools are generally used with the output of that. So with differential expression analysis, then we'll quite often get just a set of genes out of the end that have been identified as significantly differentially expressed. And then, yeah, so when we've got a set of genes that are different between the control and the treated, then we'd use that set of hopefully a few hundred to run through these tools and see if they seem to be enriched for certain biological functions. But these tools themselves don't perform any kind of differential expression analysis. They would just take some output from that. Okay, so Patrick's asking whether a similar bias dataset analysis would be beneficial for motif analysis tools such as Meme or Homer. Yeah, I think that would be really interesting to do. see what we get from those. So yes, it would be interesting. Yes, maybe someone will do it. I'm not sure that's quite on our radar at the moment, but yeah, it would be a really interesting thing to do. Okay, so I hope that's been helpful. If that's it for